Okay. So hi everyone, I'm Simal. I'm a postdoc at Luis Delicia's lab, and by the way, I brought some tools that if you guys want to see, I don't know the basically the experience with the silicon probes or some of the tools that we have been talking about. So if you just want to kind of like browse through, we have some silicon probes there, preamp in a small little box if you guys would like to see it, just to give you an idea. So my background is basically in vivo intracellular sharp electrode recording combined with extracellular recordings. I was quite fortunate to do my PhD in Neurobuzaki's lab, so I'm quite familiar with the silicon probe recordings and some kind of multi-single unit recordings and combining it with some of the LFP parameters. And in Luis's lab, I'm trying to basically build a setup where I can record multi-single unit activity in addition to the local field potential, and eventually to combine it with optical stimulation of areas of interest. So Aaron and Tom, they made really good presentations, so I will try to fill in the blanks. And the way that I prepared the presentation is just to give you an overview of the tools and the processing and analytical tools that also you will need for the analysis. And please, stop anytime that you guys want to ask a question. So before everything, let's start with a simple question. Why do we want to use this technique? Why do we care from recording using multiple site electrodes? I think both Aaron and Tom made really good comments in terms of we really need to have a good rationale to use these techniques, because any technique in neuroscience has strengths and weaknesses. So we have to understand the techniques that we are using, and if those are the techniques to best answer the particular scientific questions that we have in mind, then we should go forward with it. If not, we should kind of like try to find the most optimal technique. Okay, so in my case, I'm interested in circuit level analysis, and particularly looking at the activity of how the memory is encoded, or how the sensory information is encoded across neocortical and to some degree hippocampal circuits. So therefore, I need to have a continuous level of recordings, because we know that the same neuron behaves totally different under different behavioral states, or background activity, or sleep states, whatever you want to call it. But basically, this kind of figure nicely illustrates that if you were to sample from a neuron at any given time, and if it was happened to be three different, let's say, behavioral states, you might have wrongly deduced the assumption that this information is coming from three different neurons, whereas it is just coming from one neuron under different background information level. So that's one of the important things. The second important thing for me, since I'm interested in this synchronous activity of neurons, I need to be able to record from more than one spatial location, because otherwise it is kind of like impossible to talk about synchronous or desynchronous activity. So which already kind of like makes that I need continuous levels of recording from more than one multiple location. Now, if we really want to talk about synchrony or correlation between regions, I believe that we need two types of information. One is the unit activity, which we can get basically by extracting spikes and trying to cluster them. But the other activity, which is immensely important and as critical as unit activity, is LFP, which is basically just a summed activity over a particular region of proximal to your recording site. So I want to have this both types of information, because they are the information that is basically needed for me to be able to ask particular questions. So that's a kind of the overview of rationale. And that's also the overview of rationale why I want to use this multi-site electrodes and record wideband signal, and to be able to look at both local field potential and the unit activity. Okay, so I think I'm kind of like going a bit fast, so please, feel free to stop me. Especially if I start talking in Turkish, you should definitely stop me. That should be a good place. Okay. So this is, the way that I think that I will continue the presentation is I'm just going to give you an overview of the current tools that I'm using in the lab. And I'm going to briefly mention some of the particular components, some components that I don't really need to mention because Erin and Tom did a fantastic job. And the thing though, before I, going into detail, I want to make a disclaimer. So not all the tools here are my particular choice. So I should be, I should feel really bad if you come and, come to me like in a year from here and say that, oh, I purchased a data wave recording system because, please, I mean, some of these tools are for my, by my choice. Some of them were basically already in the lab, and I try to modify them according to the needs and basically the vision that how I want to, pursue my scientific question. So again, this is not the perfect or optimal combination. So if you are interested in particular areas, just ask me my, basically experience with them. So I'm going to start a little bit talking about electrodes. I use Neuroscience silicon probes, but there are other companies out there. But that's my preference. Then I will briefly mention about Plexon head stage. That's the head stage in the small box, if you guys want to see it. Then I will talk very briefly about the particular recording system we have, which is basically a combination of data wave, Cyworks, together with AM amplifier package. And then on top of it, I will talk briefly about the basically the processing softwares that were developed by Buzaki Lab people, and that's also the tools that I'm using in this lab. And also I want to kind of like tell you about them because they're all open source and basically free to use if you guys are interested. And finally, basically I'm not going to talk about this, but I use MATLAB for all of their analysis afterwards. Okay, so before jumping into the particular components, this is just an example of mostly raw data, on top of it some processed data that I want to show you. So this is a recording from anesthetized mice with urethane and ketamine anesthesia. And I have a recording electrode which has 2 shanks and 16 recording sites. And these are kind of tetrode-shaped recording sites. This is a silicon probe from Neuroscience. And by the colors, you can guess which sites are close to each other. And the green group are coming from one shank, which is sitting a little bit more anterior. The probe is facing the midline, and it is put in anterior-posterior direction, so that I'm sampling from, let's say, 100 micron apart different subregions within the medial prefrontal cortex, and I'm really close to the midline. And the blue and dark blue is supposed to be is coming from the shank that is 150 micron posterior. And between dark and lighter colors, there's again 150 micron apart distance. So why I'm using this particular electrode, which we will come later in terms of electrode design and how we should choose them, but I'm just going to briefly explain to you, because I am trying to stimulate basal forebrain cholinergic neurons, and I'm trying to find basically the projections, whether different cell populations in medial prefrontal cortex are activated in response to the cholinergic stimulation. In other words, I need to be able to span as many areas as I can, because we know that there is some level of topographic projections, but we are not really sure where it is. And of course, not all areas of brain are well studied as hippocampus, so we don't have necessarily knowledge of where to put the electrodes. So I need to span a wide area, and that's why I'm using an electrode with 2 different shanks. And again, the thing is, basically, I don't know necessarily what to look for. I have some ideas. I know that slow oscillations might be effective. I know that spiking of certain groups of neurons might be effective, but a priori, I don't know which ones. Therefore, I'm recording a wideband signal, which is from 0.3 hertz to 10 kilohertz. And then later, I will process to have LFB, and then I will high-pass filter and process further to have spikes, so that I can have as many information as I can. All right, so this is kind of like the raw data traces that what you see here. This is like 3 seconds of recording. And here, this is the kind of process part embedded into this raw data. This is, after the spikes are detected, extracted, and then clustered. Now I have, let's say, I don't even know how many cells here. I have some number of cells, presumably, how do you call it, single unit clusters, that I'm basically plotting the spikes of this here. And this is just to kind of like give you an idea of how, okay, so this part is raw. How this part comes is, we're going to talk a little bit about that, but this is a kind of to explain how that part comes after the spike detection, how I try to cluster cut them. But we're going to talk about them now. Okay, so now I'm going to start to go into a little bit details regarding certain components. Is there any questions or any breaks? Yeah.
You said you had 2 shanks.
Yes.
Why not 4? So if you're doing a survey experiment,
yeah. In fact, yes. This is the perfect question, and I have weird formats of tools as it is. It's primarily for budget reasons. In fact, the probes that you see here were on sale. Neuronexus have a sale, section, guys. Exactly. So these are among the tools that I have, the most suitable ones.
Black Friday soon.
Yeah, well, close to that. Close to that. Yeah. So it's like the thing is, now I'm working on my project, but until this point, it was basically setting up the systems to see whether these components are going to work or not. So initially, it wasn't really important what type of electrodes I have or which, let's say, area that I'm recording. But in fact, if you're seeing these electrodes, these are definitely not optimal ones because they are huge. They're very bulky. Especially like for mice behavior, forget about it. You have to have like really small ones. But this is the great question. This is exactly like what one needs to ask, before the project starts. And in future, I want to use these 4-shank probes, which are a bit more apart from each other, more than 150 micron, so that I can span a much bigger area. Okay. And in fact, this is the perfect transition to here. Yeah, that's perfect. Okay, so electrodes. I don't remember which one of you guys, but they made a wonderful comment. You have to really know what you want to study or what questions that you are interested in before you select your electrode. Because right now there are a variety of designs in Neuronexus, and even if you don't have the particular design of your interest, they can basically customize anything for you, for a price, of course. But basically you can get whatever you want and whatever you are interested in. So it is really crucial to know the structure and to know exactly the question that you want to answer. So I'm just going to briefly mention a couple of the parameters to keep in mind, but please, ask me questions. Before doing that, sorry, this is a kind of a very bad resolution, but this figure is supposed to show, this is again a recording from Buzaki Lab, from Fujisawa. It's also in the Neuronexus website, so you can get it from there. Basically, these are the shanks of the silicon probe, and in initial staining, it would be very even hard to kind of make up where the track was. This is, again, by no means to say that there is no damage. There is definitely damage. Or whether that damage is, at a level that is, I don't even say optimal, acceptable or not, even that's a question. But still, it is not like, I don't know, inserting, let's say, cannulas or some other things that we do and, we still get some behavioral readouts from them. I do that too. I use sometimes these +6 cannulas for, optical stimulations, and they are humongous compared to this, and they definitely leave some damage. But yeah, if you don't put dye or some kind of a marker, it's often the case that if the surgery is good, the experimenter is very talented or experienced, then you may not even really recover the track where you recorded from. That was just like 2 cents on this. Now about design. So of course, there are a couple important things. The first thing is, I guess, to keep in mind, where is your region, how deep it is. So that, this whole thing is called shank, so that you have to know the length of the shank in terms of where you want to reach in the brain. Then the second important thing is how many channels you want to record from. which is usually not by choice how many channels you want to record from, but you're limited with the number of recording channels in your setup. If you have a channel, system for 32 channels, obviously you can't record from 34. But it doesn't mean that you have to record from all 32. You can always record from 16 channels and ground the other 16 channels so that you don't have much noise. Or better, you can use 2 silicon probes from perhaps in different regions of the brain and record from all 32 places. The third important criteria is what type of a configuration you want from your recording sites. For example, these tetrode type recordings are very nice in terms of unit isolation and desirable, especially if you want to record from relatively nicely packed neurons and if you're really interested in clustering them into single units. Whereas if you're interested in depth profile, you might want to have something in a more linear extent, expanding basically a certain area in dorsoventral axis. The second important thing is, and in fact, you can also see some even more fancier type of designs where you can have even more information from a within spatial resolution. Another important thing, and which Aaron kind of mentioned, so I'm not going to spend much time on it, you can now choose this recording sites. These are iridium, recording sites with relatively high impedance. The impedance might change around 1 to 3 mega ohms, but you can basically define how big of an area that you are interested in. In a very simple way, the larger the area, you are more prone to record the local field potential. The smaller the area, you are more prone to record reliably unit activity. If you choose something in between, you can usually get, both levels of activity in a reliable way. So, any questions about this? Yeah.
Do you try electroplating, for example, with platinum black?
On silicon probes, no.
Why not?
Probably I would break them because these are like super tiny things. I think nowadays you can ask them to do it for you. The thing is though, you can do it if you want to do it, but I wouldn't try to kind of do it myself. But you would definitely need a very good microscope and some really handy tools, I think, if you want to do anything with the silicon probes, because they are very, very fragile. It's very easy to break the shanks, basically.
What's the point? Excuse me? Reducing impedance. Then you can have small electrodes, which give you a better definition of the cells.
It essentially increases the surface area by adding all these small ripples.
It's cauliflower.
Yeah, so it lowers the impedance by increasing the surface area without drastically changing the geometry.
You should be able to do it just in a liquid. Like you could be able, maybe you could, I don't know how else you would do it, but you can dip the tip of the electrode in there and then manually go through and apply current to every site. It would be tedious.
It would be. I would probably not take the chance of trying it, unless at least we have much bigger grant money, but I would give emotional support.
How much does one probe cost, just so we have a sense?
Yes, so that's a good question. So if you buy something standardized, and now they have a huge array of standardized probes, you might get something, especially something like the way that I have, like really bulky. These are called acute probes. You can get them like $200 to $300, depending on the recording size, depending on how big it is. But
that's on sale or not on sale?
This is not on sale. On sale, you can go even cheaper, and I can kind of like give you guys I think nowadays they have this inventory sale going on. So if you guys are interested, inventory sale, you might have probes with some defective sites, but at least you know them a priori. You might have also probes with no defective sites that it's just that they are not, doing any more of them. So.
And you can reuse them?
Yes. So depends in fact. So if you are doing an acute recording, so that doesn't have a shank anymore. That's why that's on display. I should have told you that. In the box, there are probes with shanks. Sorry, yeah. Yeah, so if you guys can, pass the box. So before even answering that question about the money thing, unfortunately, if you're interested in mice physiology and if you're interested in, let's say, rather uncanonical areas, then you might have to ask for more customized probes or less standardized probes. They might be like 400, 500.
Yeah. And how many times can you reuse this?
Yes, so if you are doing an acute preparation, meaning that, it's on a holder, it's flowing somewhere, if you have seen my thing. And if you're very careful, if you don't break it, you can use it 20, 30 times.
Oh, wow.
If you don't, as long as you don't break it. And after each surgery, you can kind of like try to clean it a little bit with water. In past, we use a little bit dextrose, but water is enough. So you can definitely reuse it. Now, if you implant it on an animal, it's already a huge success that you implanted it without breaking it. So you can always go crazy to try to take it out in the end of the surgery, but honestly, I've done that. I sometimes, in 2 occasions, I took out the probe. It seemed okay, but I would never trust it to implant in, new animal because it's a huge investment too. So for chronic experiments, I can say that it's probably one animal. If you get like one animal, you're lucky. Let me put it this way. Okay, so I think we can move on the head stage, and I'm gonna be kind of like try to go fast because I think we don't have much time. So Erin made a perfect point about noise. Now the thing is though, since these recordings are relatively capricious compared to, let's say, single wires or tungsten electrodes, you want to basically capture the signal as close as possible to the source and differentiate from your reference signal and amplify it. That's why we need devices called head stages or preamps or whatever you want to call. The circuit is in a way that, again, it depends on the recording channels that you are recording from. But usually you have, let's say for 32 channels, you have 34 channels of an amplifier with some gain. That gain is usually 20x these days. So that you can wire recording channels to this 20x gain amplifiers, and you have optional, let's say 2 sides or sometimes 1 side for reference from a de-insulated electrode. It can be a skull screw that I place it on cerebellum if I'm recording from basically anything frontal to cerebellum or vice versa, and you can feed that signal. And in addition to that, often you have 1 or 2 buffered signals where, again, there's an amplifier, but it is not basically amplified. It's just the gain factor at 1x. So there is this, this little circuit. Unfortunately, if you want to record from these electrodes, you need this. It doesn't have to be from Plexon. There are many other head stage companies out there, but you need something like that. And the important thing is, perhaps I should make it. So in the other box, for example, you see some connectors. The critical thing is you have to interface your silicone probe to this device. Okay, so this is the input area. So again, if you have a nice amount of grant money, you can buy things already matching nicely so that you get the silicone probe with the connector connected, and then you can just plug them. If not, you can try to wire them. It is easy to wire those with the probes that you kind of like see here, because they have blunt, very big sides that you can basically just solder. But if it is a smaller, more delicate, silicone probe, then it becomes more problematic. And also it becomes more problematic because they're changing these connectors to this double row, really tiny ones. Then it becomes like really hard to solder it. But basically you have to interface the two in some way. And you have to interface the preamp to your recording setup also in some matching way. Now this part is, I think, can take hours. I'm not going to go into much detail. The thing is, though, you have to, probably this is the most critical decision. If you are interested in having a system, having a recording setup that can record from multi-site electrodes using, let's say, this relatively high impedance electrodes, you have to really first choose your recording setup very wisely. Now, in Buzaki Lab, I have used RC electronics for years, and I was super happy with that. In a way, it was super, basic recording setup that you would just continuously stream the data, and that was basically it. It would just digitize and store it. Neurolinks was another setup that was primarily used in Buzaki Lab, and people were also very happy about it. I never used it, but I also trust that that's a good setup. What we have here in this lab is data wave. There are goods and bads about that, but honestly, if I were to buy a system for my lab, I wouldn't buy that one, for example. It wouldn't be my choice. So it is really critical that you have good sense of what you want to do, and then find someone that uses similar type of recording setups and get first-hand knowledge from them, because everything else has to fit together. And there are certain things that we don't really discuss. For example, certain probes don't necessarily match with certain head stages, and then head stages don't necessarily match with recording systems. So you have to make sure that you start from the recording system and go from there. And I think we can also, I don't know, maybe have a nice, fun beer session on super user-friendly systems. I honestly don't like them. I hate them. But there is this motto that now they kind of sell you a box and it can do everything. I have super bias about that. Honestly, in terms of the recording setup, I like to have the most primitive thing. That if you want, you can just, with a BNC, you can get the signal and check it in an independent source. You can listen, so on and so forth. I don't necessarily like that setups that can do all.
What are some of the makers that are more?
For example, data wave is do it all.
Okay.
The thing is, maybe I should rephrase what I said. Neurolinks also can do it all, but Neurolinks can also provide you simple formats.
Right.
So yeah.
Just specifically to that, I agree with you. It's really great to be able to sort of check your work and pull a signal off and listen to it in the analog output. And Neurolinks, because it digitizes things really early, I and other folks I know have run into problems where they haven't been able to get a nice low latency analog signal.
Oh, I didn't know that.
So I think they're kind of working on it, and they're plugged into that. And one other company I would put on that list is Tucker Davis.
Oh, Tucker Davis.
And they're kind of more you roll it yourself in terms of the interface.
Roll it
yourself. That's not a thing. And it's more flex. You in particular might want to look at it because it's more about you kind of set it up the way you want it, but it is less. It's definitely less user friendly. But it's very much kind of it's kind of a more modern platform in that it's very low. They put a big premium on having things be really low latency, and it's got built-in tools to build little. It's got some visual programming language that allows you to sort of build simple real-time applications. And then I think there are some open source hardware projects that variously come and go, and some of them I'm not really actually aware of any that are fully fledged and fully working, but I'm sure. I agree this would make its own nice hour discussion.
I can just say that from the molecular physiology side, in my lab here in Moore's lab, we use Python systems, and in Krishna Shinoi's lab, they use BlackRock systems, which are just two other options, and both of our labs are really happy with it.
So this is awesome to think. I think to hear the people's opinion that people actually using it, I think this is awesome. So this is a great reference. Okay, so I'm going to move away from this topic, which obviously I'm not very keen to talk more into. Anyhow, so let's now talk about a little bit with what to do with the data. Because the good side and also the bad side is now you end up this humongous amount of data. It can be, depending on the recording side, depending on the hours of recording, you can have up to, 10 gigs of data. from one day. So what you're going to do with that. And especially if this data is wideband, now even before starting to analyze your data, you have to process it somehow. Now this is, I think, the second critical decision if you are setting a system. You really have to know a priori, or at least you should decide a priori what you're going to do once you recorded the data. So what are the options out there? We just mentioned that there are certain systems and more companies that are basically offering you the tools to use from their own rather commercial softwares. Some of them, I think you can really trust some of them, because some of the companies that have been out there for a while, they worked really closely with scientists to kind of develop these methods. So I'm pretty sure they have some kind of decent level of processing. If you're coming from an electrophysiology lab which has, let's say, certain level of history in that field, they often have their own custom softwares and processing tools and processing scripts. So that's another option. Now, if you're starting brand new, you can either write your own tools, which I don't know how many years that it can increase the life of postdocs and graduate students, or you can choose like one of these commercial companies, or you can use already developed tools from some of the let's say established labs. So now today I'm just going to talk about this particular combination from Buzaki lab, because basically I was using these tools when I worked there, and now instead of starting something from scratch, I try to use their own tools. And they are open. They are developed by scientists in the lab, and it has been growing number of labs starting to use them. So if you guys are interested, I can give you some more information, and you can always contact them, and they are super happy to share their softwares and also brainstorm and to receive inputs from basically other scientists. So, okay, let's go into them like very briefly, because I think we're really out of time. Okay, so one thing, the first tool that I use as soon as I record the data is called Neuroscope. It is basically a graphical interface that you can just stream the data. And I like to do that because first I want to have an idea of what my data looks like. If you are particularly working in behaving animals, you might have this like periods of super artificial noise. You might have periods of cables unplugging. Even if you are doing anesthetized recordings, you might have basically periods of mayhem, or you might have perhaps something really funky that you haven't catch during the recording. So I just browse the data, and Neuroscope enables basically just raw data continuous browsing. That's good. Now on top of that, I use a software called ND Manager. Nowadays they kind of like lump them together, so it is just under the name of Neuroscope. It basically gives you in a package format processing scripts that you can run to low pass, high pass, extract spikes based on the particular thresholding parameters that you provide, and then use a kind of automatic clustering algorithm to extract features, and even to run PCA and to give you this, let's say, initial clusters of data.
Simal, is this on what platform, and does it run in MATLAB, or is it?
So that's an important one. So all of these tools are primarily developed on Linux, but most of them are compatible to run in Mac. You can, I believe, also you can run some of the things on Windows if you have virtual,
Gotcha.
what is that called? I can't remember.
Virtual.
Yes. So yeah, there is some level of operating system requirement that one needs to keep in mind. Okay, so this is just a kind of display of the data. Was there a question? No.
It's actively being developed, that software?
Yes,
so
exactly. So that's the nice part of it. So they have, this basically, now here comes the sideline story. So Lynn was, in fact, wife of one of the postdocs in the lab. She was also a postdoc in the lab. But that postdoc is Mikhail Zgoro. Right now he's in Collège de France, and he's, you know, a primary investigator of his own. So they kind of like built together. One of the people who had primarily inputted was Joseph Cicichvari. You know, he's in Oxford. So all of these people who kind of like use these tools and who develop these use are still using them. And in fact, the more that they kind of, let's say, grow up and move and branch out, the number of labs are kind of increasing in number.
They're all contributing back to the central repository.
Yes, yes. Yes. Probably I am like, so far I am the least contributing and probably receiving, but you know. In fact, we should talk about a little bit in terms of getting back. Because getting back also depends on, you know, how similar or dissimilar your recording setup is and your region of interest. And we're going to come to that about the, because, okay, before I forget, it is important to keep in mind that, you know, how these tools are developed, or by whom. And by whom, I mean people with what type of scientific interests. For example, most of these tools were developed by people who have been recording in hippocampus in rats. So, you know, I think someone from here mentioned this earlier. Hippocampal recordings in rats might be something very different if you are interested in layer 2, 3 in mice. So, you know, you have to keep in mind, and basically in terms of giving back, I think that becomes critical along that aspect to kind of expand the tools. Okay, I think, yeah, we were here. All right, so the last two ends of this softwares or tools are ClusterQuick and clusters. So ClusterQuick is basically the automatic clustering algorithm that looks at the PCA, basically principal component analysis, and kind of like initially gives you a number of clusters. In a way suggesting that these are the optimal cluster cuttings that the automatic clustering can, you know, develop.
So
now, yeah.
Just a step back, so clustering refers to taking spike waveforms and separating them so you know one neuron from another.
Yes, exactly, exactly. And maybe I should explain it a little bit. Okay, so let's say you have the wideband signal, you high-pass filtered it, and then so you put some thresholding requirements, and even before that, you know, you kind of calculated the root mean square of the basically the continuous signal. Then you put some thresholding, I don't know, plus minus 3, 4 standard deviation, and then you detected these time points whenever you know, your signal kind of crossed that threshold. And once you have this information, let's say let's say detection, then you go there and upsample. Let's say 1 millisecond before and after of that period, so that you can have this waveform in a much more you know, high sampling rate. And then on top of that, you extract features by using, you know, PCA, so that you have a much better idea about the waveform. Not just amplitude, let's say, but also some other characteristics of the waveform. And then you can put this to the automatic clustering algorithms. Okay, so ClusterQuick does that. Then you have another very strong tool and easy to use is clusters. Clusters is basically the graphical interface that you can upload this you know, outcome of the ClusterQuick. Let's say you have 20 number of clusters, and you can look at them in the basically the, you know, principal component space, and you can look at them in their waveforms. You can look at them autocolorograms, cross-colorograms, error matrix that to some degree tries to tell you that, oh, maybe this two should be splitted, maybe this two should be merged, maybe this should be splitted, so on and so forth. And then you can draw lines manually and modify the output of the automatic cluster. And now I have like a couple examples, for example, to show. So, you know, remember the earlier traces that I showed you with, you know, 4 tetrode recordings, greens and blues. So I'm just going to show you some, you know, clustering outcomes from those traces. So this was one of the tetrode sites. I only had, you know, one reliable isolated unit. And when I say isolated, keep in mind that there's always, and there is always going to be an error factor in it. So, you know, now this is again a philosophical discussion, but we are never even sampling the whole spikes to begin with. So there is definitely an error. And this error is sometimes larger, sometimes smaller. And maybe at some point we should have discussion about this as well. But this is kind of like relatively clean unit because the refractory period has some noise, but that noise is, you know, lower than an asymptotic value. So that, let's say I would trust this, you know, as being a single unit. And you can see the waveform, and in the EBAE space, you can basically see the first components of the second and third electrode site, and you can have some, let's say, degree of trust. Now, if you have more than, you know, one cells or spikes of cells, then things might get a little bit more complicated. So this is from another tetrode site from the same recording. And basically, you know, the waveform, the colors of the waveforms correspond to the basically the spikes that you see in space, and also the colors of the autocorograms here. So, you know, if you look at it, not all of them are, you know, great cells, and not all of them I would, let's say, how do you call it, include in certain levels of analysis. For example, this is, you know, to begin with, even though it has a, you know, beautiful waveform, it has a very low number of spikes, so probably I wouldn't consider it, at least by itself. I would perhaps combine it with other units and use it as multi-unit, but, you know, I wouldn't use it as a single unit. Another example. Okay, so here, for example, you know, this is, again, a nice cell, but, you know, it is not an isolated unit. And I know that because, you know, it's very crowded in the refractory period. This is, for example, a very nice cell. This is a nice cell. Now, you know, if you know, I guess this is a nice part to look at the space here. For example, it is very easy to, you know, isolate this blue guy, which has also very distinct waveform. Whereas it becomes more problematic to isolate, you know, these guys. They have all similar waveforms, especially if you were to just look at one recording site. Now, this is also another example to show that the more spatial information you have regarding a neuron, it becomes easier to separate them. Because imagine that if we were just recording from site 11, then, I mean, it would be almost impossible to isolate neurons. Okay. So, I'm going to try to close very quickly. So, I'm not going to talk about this paper, but for you guys who are interested in cluster cutting, read this. This is a very fun paper where the people in the lab kind of clustered neurons, and then they checked the verification of their clustering because they had a couple of recordings where they recorded simultaneously from the same cell, intracellularly and extracellularly. And I guess from the initials you can make up who is who, so it's a very, very interesting one. The bottom line is there's some level of error. And people have certain biases to make certain errors. At least in this particular lab, these particular people were more biased towards false exclusion of spikes, meaning that some spikes were to be, you know, emitted from the cell, but they were more prone to basically discard them. So that's important. And for all of us, especially the mice people, I think it is important to keep in mind that you know, low amplitude spikes are really disfavored by many of the, you know, clustering algorithms out there. So we have to be really careful. The other thing is, if there is synchronous unit activity, such as in slow oscillations and from a very confined space, then it becomes super hard to kind of, you know, separate those. Now, why do we need them? Because let's say that you have done a manipulation and you want to see the effects of it. Then you can record, you know, spike histograms of each individual cell, and you can look at whether you see an effect or not. So it gives you more, you know, basically levels of power to look at, and more data points. Another thing, I'm not going to go into detail, but if you record simultaneously and if you can nicely isolate these cells, you can look at the interaction among the cell pairs. Monosynaptic connections, and then you can even ask more fancy questions. Okay, the manipulation that I do, whether does it change the direct communication between two cells? Okay, issues that needs debugging. Definitely related to the softwares, you have to make sure that you have the right operating systems that can work. You have to ensure the silicon probes and design and whether how they are compatible together. Noise, not even need to mention. The second thing is initially for people who might just start doing this, the wiring becomes a bit problematic, and if you need help, I'm happy to help, because basically these tools come with maps, and I think I have some here. And you have these maps from the preamps, you have the maps from the silicon probes. You have to ensure that how those two talk. And finally, there are even more areas that kind of like make you go crazy. But I guess we just have to embrace them. So thank you. I'm sorry, I think.